expression. So this mastery series, this is a rapid revision series. So we'll be revising all the important topics and high yield questions so that we can assure you top-notch grades in your university exams. So for today's class, we'll be talking about regulation of gene expression under the heading of lac operon. So this is very important. Then followed by regulation of expression in eukaryotes, and we'll also be discussing the various types of genes. Reintroducing you to the core features of Auricle. Here at Auricle, we have live classes to elevate your learning experience. We also provide you with recorded lectures so that you can assess your subjects anytime, anywhere, as per your convenience. Then we also give you notes based on the latest CBME syllabus, followed by MCQs so that you're able to assess your knowledge. Then here at Auricle, we have 3 different subscription plans available. The basic, which is for 3 months and comes at 15,000. The premium plan, which is for 6 months. Sorry, the standard plan, which is for 6 months and comes at 30,000. And then the mentorship plan, which is for 12 months and comes in 50,000. So in the mentorship plan, we also give you live doubt clarification sessions, followed by one-to-one personalized guidance, and notes will also be provided. Then a very unique feature of Auricle is that you have the availability of personalized counseling sessions. So if apart from the academic support, you need any extra support from our end, feel free to book a slot with us anytime. Okay, so with this little details about Auricle, let's continue. So I'll be quickly taking DNA repair, which is a very important topic. Yesterday, because of time limitation, I was not able to complete that. So I'll quickly take that up. Then we'll begin with gene regulation or regulation of gene expression. Okay. Okay, so while we were talking about replication, I told you that DNA polymerase, apart from having this polymerase activity, it also has proofreading activity. So while you were replicating or synthesizing your DNA, copying your DNA, the proofreading mechanism was already active to ensure that the DNA is replicated or copied with a very high level of accuracy. Okay, this accuracy is very important because it is required to maintain the genomic integrity and to prevent mutations. But what happens in spite of all this proofreading and in spite of all the measurements that you're taking to ensure this fidelity, what happens sometimes there can be a mismatch or there might be mutations taking place because of there can be many reasons like your ionizing radiations, various chemicals, exposure to UV rays and all that. So they can cause DNA damage. Okay, so what happens now? You have to repair these damages in order that correct genome is transferred to the next generation. Okay, so therefore that is where your repair mechanisms comes into action. So apart from proofreading, you have DNA repair enzymes which are constantly scanning your DNA that is being synthesized to look for damages. So inbuilt cellular mechanisms to correct damage or errors in the DNA structure, that is what comes under DNA repair. Now, it is essential for maintaining genomic integrity and preventing mutation. So you have DNA repair enzymes that are scanning the genome that is being synthesized in order to check for any damages. So you have different repair mechanisms. Let's talk about them. So the first one is base excision repair. Then you have nucleotide excision repair. Then mismatch repair and double-stranded breaks. So in base excision, here the description says, it corrects small DNA lesions such as spontaneous depurination. Specific DNA glycosylases recognize damaged bases, excise them, and create an A basic site. Then endonucleases remove the damaged portion and DNA polymerase ligase fill and seal the gap. So in DNA excision repair, what is happening? Let's suppose, okay, here you can see this diagram here. This is the normal DNA. Now you can see in the normal DNA, you have T, C, C, and T region. Okay, so what happens because of spontaneous deamination? This C here because of deamination, it changes to uracil. You can see here. So this is the defective GNA. So what is happening? And this is a type of a mutation. Okay. So because of spontaneous mutation, deep urination or deamination, what you can say your cytosine is changing into uracil. So this is not a mismatch. Mismatch, it was correctly paired, but what happens because of mutations, this cytosine changes into uracil. Now it becomes a mismatch. Now what happens? This is defective DNA. What is going to happen now? Glycosylases, in this case specifically uracil DNA glycosylase, it is going to come into action and it is going to create an A basic site. That is, it is going to cut the glycosidic bond. Okay. So it cuts the glycosidic bond and not the phosphodiester bond. So what is this glycosidic bond is present between the sugar and the base. Okay. So your base is going to be removed. What is going to remain here? Still your phosphate and sugar is still going to remain. So a base is removed, only uracil is removed and an A basic site has been introduced. Now what happens? Endonucleases will come and that it will remove that sugar and phosphate as well, and it will create this gap. Okay, now this gap is going to be filled by DNA polymerase, and then the neck is going to be sealed by DNA ligase. So this is the mechanism of base excision repair. So it takes place when there is spontaneous deep urination. Okay, so this is not a mismatch. Now, next type of repair mechanism is nucleotide excision repair. Here, what is happening? Repairs a broader range of DNA damage, including those caused by UV light or ionizing radiation. A complex series of steps involve recognition, incision, excision, and repair synthesis to replace the damaged DNA. So, in nucleotide excision repair, what is going to happen? There are going to be, because of the UV light or ionizing radiations, there are going to be damages like your pyrimidine dimer. Pyrimidine dimer formation. Okay, now what is going to happen? Because of this, here what enzymes are involved? Okay, so here the enzymes that are involved, they are known as ABC nucleases. Okay, so these are three subunit. They are enzymes which contain three subunits. That is, they are a product of three different genes: UVRA, UVRB, and UVRC. Now, what happens? It is going to they are going to come into action, and they are going to remove a small oligonucleotide that is from the lesion on both the sides of the lesion. For example, let's suppose this is your DNA. And what happens? There is a pyrimidine dimer formation. Okay. So now your ABC exonucleases, they're going to come and they are going to remove a small nucleotide like this on either side of the lesion. And then it also has helicase activity. Unwinding is going to take place. And then you can see this gap is formed. Okay. Now this gap, it is going to be filled by DNA polymerase and then ligase is going to seal the nick. So the basic mechanism is same, that excision is taking place, a small segment is being removed and then DNA polymerase is going to fill up the gap and then ligase is going to seal that. So this is like the basic mechanism in almost all the repairs. So that was about nucleotide excision repair. So here you're dealing with bulky adducts like pyrimidine dimers, and the key enzyme in this case is ABC exonuclease. Okay, and then a very important disorder associated with this is xeroderma pigmentosa and Cockayne syndrome. So remember that. Then talking about mismatch repair, in mismatch, what is happening? That there is some mismatching, incorrect base has been matched. Okay, so what happens? Your newly synthesized, your parental DNA, it is having methylation, and your newly synthesized DNA is not methylated. Okay, so at every 1,000 nucleotide, there is a GATC sequence. Okay, so in parental strand, that GATC sequence is going to be methylated, but in the newly synthesized DNA, that methylation still not there. Okay, so there is a little lag or delay between replication and methylation. So that way your mismatch repair enzymes, they're scanning the newly synthesized DNA. So they're able to differentiate between the parental and the newly synthesized DNA. So if they see any mismatching, then again they're going to recognize this mismatch, make a cut between the two GATC sequences, remove that nucleotide sequence, then DNA polymerase is going to fill the gap correctly, and then ligase is going to seal the nick. Then talking about double-stranded breaks, here what is happening? It is going to be something like this. Okay, like this is your DNA. So they are going to be double-stranded breaks, as you can see here. So in double-stranded breaks, it addresses breaks in both strands of the DNA, preventing harmful consequences like chromosomal translocation. So it is important that you repair these double-stranded breaks. Otherwise, it will lead to chromosomal translocation. Repaired through homologous recombination or non-homologous end joining. So one mechanism of repairing this is non-homologous end joining, and the other one is homologous side-directed recombination. Okay, so it depends upon the cell that out of the two mechanisms, which one it is going to take. Whether it is going to be through non-homologous end joining or it is going to be through homologous recombination. So that was about double-stranded breaks. And this non-homologous end joining, it is error-prone, okay? Whereas the other mechanism, it is less error-prone. But it depends upon the stage of the cell, its presence of the cell in the cell cycle, at which stage it is going to be present. So your non-homologous end joining is going to take place throughout the cell cycle, but it is more predominant in the G1 and the G2 phase, okay? Whereas your homologous recombination, it is more predominant in the S phase, okay? So that was about double-stranded breaks. Now I think we've covered with DNA repair. Now let's move on to gene expression. Okay, so let's begin with gene expression or regulation of gene expression. So introduction to gene expression, your gene express themselves through the synthesis of proteins, a process known as gene expression. So DNA itself is not expressive. It is through forming RNA, that is through transcription, and then that RNA being translated into protein. That process is known as gene expression. Okay, so your transcription and translation, that comes under gene expression. So now, while all cells share the same set of genes, not all genes within a cell are active, okay? Now, what it means is that genes are present in each cell of the body, okay, and are same. Like in the pancreas and the liver, the same set of genes are present. But in the pancreas, insulin is being secreted and the liver is not secreting insulin. So what is the reason behind that? Because in the pancreas, the gene for insulin is active, whereas the liver is also having the gene for insulin, but in the liver, it is not active. Therefore, your liver is not producing insulin. So that is the basic difference. Therefore, this is what this point here, second point explains, that while all cells share the same set of genes, not all genes within a cell are active. And the example like I just gave you, that your pancreas, they have also the gene for insulin and which is active, therefore insulin is being produced. And the liver, the insulin gene is not active, so the gene is present but insulin is not being produced. Okay, now gene expression is regulated, leading to two main types of genes: constitutive genes expressed consistently and regulated genes which are activated under specific conditions. So you have two types of genes, that is constitutive genes. These are those genes that are expressed all the time. For example, your hexokinase. Whereas your regulated genes or inducible genes, they are those genes that are induced under a certain circumstance. For example, your glucokinase, which is induced in postprandial state. Okay, so we'll talk a little bit about types of genes, that is constitutive genes. Like I just told you, these are genes consistently expressed in all cells. Characteristics: they are unregulated and essential for basic cellular functions. So example, enzymes involved in glycolysis. So your glycolytic enzymes, they are constitutive genes because your glycolysis needs to take place at all times. Now functions: it provide fundamental cellular activities under normal conditions. We're talking about Regulated genes or inducible genes. These are those genes whose expression is controlled by regulatory elements. Characteristics: it is responsive to inducers or repressors expressed selectively. Then examples: genes regulated by insulin responding to specific conditions. And the functions: they are adaptable gene expression allowing cells to respond to changing environments. So this will be more clear, the regulated genes, once we are going to talk about lac operon. So that is what we're going to study today. So talking about lac operon. So what is lac operon? So your structural genes along with the regulatory genes, they constitute an operon. Okay, so your operon is the best model that is studied for gene expression. Okay, so regulation of gene expression, it can be studied well by understanding the operon model. So your lac operon model in E. coli, that is how your E. coli is utilizing lactose. That is what lac and lac operon means. Lactose, okay, lactose operon. That is what we're going to study in order to understand regulation of gene expression. Okay, so the lac operon is a gene regulatory system found in prokaryotic cells, particularly in E. coli bacteria. It consists of structural genes, a promoter, an operator, and a regulatory gene. So that is what I said. Your structural genes along with their regulatory genes, they constitute an operon. Now the components of lac operon. So you have the structural genes, which are Z, Y, and A. Then these Z, Y, and A, they encode enzymes involved in lactose metabolism. So your gene Z, it is basically beta galactosidase, and it breaks lactose into glucose and galactose. So it is basically involved in lactose metabolism. Then you have permease, which facilitates lactose entry into the cell. So permease is going to allow large amount of lactose to enter into the cell, and the function of transacetylase, it is still not known. So here in this diagram, you can see these are the structural genes here. Gene Z, gene Y, and gene A. So this one is beta galactosidase. This is permease, and this is transacetylase. So these structural genes, that is gene Z, gene Y, and gene A, these are the structural genes. And when these genes are active, what is going to happen? Your lactose is going to be utilized, or it is going to be metabolized. Okay. Now, one very important thing is that for E. coli, the preferred source, metabolic fuel, okay, it is glucose. Glucose is the preferred metabolic fuel. Okay, what does this mean? That in a medium, if you put E. coli in a medium where glucose and lactose both are present, it is going to first utilize glucose. It is going to grow, and after some time you will see when the entire glucose has been used up, the growth is going to stop, and then it is going to again start utilizing lactose and again start growing. So then doctors try to study, or the researchers try to study what this switching, that how initially in the presence of glucose, the enzymes which were supposed to utilize or metabolize lactose were off, and then what is going to happen, or what happens basically, that in the absence of glucose, the enzymes that are responsible for lactose catabolism or metabolism, they are switched on. So this switching on and off of the enzymes responsible for lactose metabolism, that is what is regulation of gene expression. So how is E. coli regulating the expression of those genes which are responsible for producing enzymes to utilize lactose? This was studied with the lac operon model, and that is exactly what we are going to study. Okay, so you have these structural genes. Okay, now what do you have? You have this promoter. So to the promoter region, we all know the promoter region is the region where your RNA polymerase enter. So this is the promoter region, and your RNA polymerase is going to enter through the promoter region, and then it is going to transcribe these structural genes. That is what is happening. But now we are saying that these structural genes, they are having regulatory genes. Okay. So downstream of the promoter, you have this region, which is known as your repressor region. Okay. Or the operator region. And upstream of the promoter, you have another region which is known as cap region. And then you have another gene known as LAC-I, which is producing a protein which is known as repressor protein. And this repressor protein, you can see this is the repressor protein in yellow that it is producing. And this repressor protein is binding to this operator region. And this repressor protein, LAC-I, is constitutive. It is a constitutive gene. That is, it is expressed all the time. So now I think the picture is somewhat clear to you. I'll explain this again, that what are all the regulatory and structural genes present in the lac operon. You have three structural genes, that is gene Z, gene Y, and gene A, as shown here. This, okay? You have three structural genes. So when these three structural genes, they are expressing or they are transcribed, they are producing enzymes which are responsible for metabolism of lactose. So when these enzymes, these structural genes, they are active, they are on, then the E. coli is able to use lactose. Okay, that is one point that I've told you. Now what I'm saying, that you have the promoter region. In transcription, we've all studied that the promoter is the point from where the RNA polymerase is going to enter the gene, and then it is going to transcribe the structural genes. Now, this promoter and this RNA polymerase, they are under strict regulation. They're under strict regulation of operator region. So this operator region is a region downstream of the promoter, as you can see here, and a cap region, which is upstream of the promoter. Okay. So now this promoter has a downstream region known as operator region and an upstream region known as cap region. Now what happens? This RNA polymerase is going to transcribe the structural genes only when it is stimulated by a cap protein present in the cap region. So this cap protein present in the cap region is going to act as a positive regulator of RNA polymerase, and there's another repressor protein present in the operator region. So this repressor protein present in the operator region, it is going to act as a negative regulator of RNA polymerase in the promoter region. Okay. So now what I'm trying to say, see, I'll draw another diagram here, a handmade diagram which is going to simplify things for you. Okay, the structural genes present are your Z, Y, and A. And then I'm saying there is this promoter region. In this promoter region, what do you have? RNA polymerase. And then you have this operator region. Operator region. And then you have a cap region. Okay. Now something very interesting. In this operator region, now see, remember this now. So there is this person, you can understand, okay, in the cap region, there's this one person which is known as cap protein. And this is giving a push. Okay. So take this push as a positive regulator or as a stimulator. Okay. And then here in the operator region, you have repressor protein. take this repressor protein, as you can see, it is pushing in the opposite direction. So it is stopping the RNA polymerase in the promoter region. So this is a negative regulator. Now I think it's very easy for you to understand. Now for this RNA polymerase to work, it needs two conditions. Firstly, this repressor protein, that is something which is stopping it to transcribe the genes, that should be removed. And also this push from behind should be present. That is cap protein should be present in the cap region. Okay. So that is the entire regulation process. Now see how it's going to take place. Now there is, as I already said, there's another gene known as LAC-I, which is a constitutive gene, and this is producing the repressor protein. So the repressor protein produced by this LAC-I, this is binding at the operator site. Now what happens when glucose is present in the medium? So glucose is going to inhibit adenylyl cyclase. Okay. So when glucose is present in the medium, glucose present, it is going to inhibit adenylyl cyclase. Now your adenylyl cyclase, it was converting ATP into cyclic AMP. So since it is inhibited when glucose is present, what will happen? Your cyclic AMP levels will decrease. And when cyclic AMP levels decrease, your cyclic AMP was basically responsible for activating cap protein. Okay. So cyclic AMP cap complex was going to bind to the cap region. So, but the cyclic AMP level has decreased. So what is going to happen? Your cap is not going to bind. Cap is not going to bind to the cap region. So that push is absent, okay? And since lactose is not there in the medium, so what is going to happen? When lactose is present in the medium, some amount of it is going to enter the cell and it is going to change into allolactose. Now this allolactose is able to bind to the repressor protein, which is already bound to the operator site, and it is able to put that repressor protein, making a conformational change in it from its operator site. So that repressor protein, which was obstructing your RNA polymerase, that was removed. But here what is happening? Lactose is not there. So repressor protein will not be removed from its place, and your push, that is the cap protein, is not present. Therefore, what is happening? Your structural genes, they are not being transcribed. Okay, so your RNA polymerase is not transcribing your structural genes. So that was the scenario in case when glucose is present and lactose is absent in the medium. Now, another scenario can be glucose is absent and lactose is present. So when glucose is absent and lactose is present, the E. coli is supposed to utilize your lactose. Now, what happens? That is your structural genes should be active. So how are they going to be active? They need that push from the cap protein, and that obstruction from the repressor protein should be removed. So what is happening when glucose is not there in the medium? So your adenylyl cyclase is going to be active. So your cyclic AMP level is going to increase, and cyclic AMP is going to bind to your cap protein, and the cyclic AMP bound cap protein is going to bind to the cap region, and it is going to push the RNA polymerase to transcribe the structural gene. And also what is happening, since your lactose is present in the medium, some of it is going to enter the cell, change into allolactose. Allolactose is going to pull off the repressor protein from the operator region. So repressor protein has been removed. That push from the cap protein is present, and the structural genes for lactose, they are going to be transcribed, and then your lactose is going to be utilized. So that was the case when lactose is present and glucose is absent. Now, there can be a third scenario where glucose is present and lactose is also present. So initially, what is going to happen? Your lac operon is going to be switched off. That is because of the presence of the glucose, your cap protein, that push is not going to be present because your adenylyl cyclase is going to be decreased. Okay. And your allolactose is going to be present. The repressor protein has been removed. Okay. When in the case when both glucose and lactose are present, although the repressor protein has been removed, but that push is absent. So what is going to happen? Your lactose enzymes are not going to be formed. That is, your structural genes are not going to be transcribed. Okay. So once all the glucose has been utilized, now what is happening? That push will also be present because the level of adenylyl cyclase will increase. And then what will happen? The switching will take place. Okay. After the glucose has been used completely, now the E. coli is going to utilize lactose. So we've spoken about all the three scenarios. We've spoken about all the genes. Now, since I've explained to you everything, we'll read the notes, so it will be more clear to you. So, firstly, talking about the components of lactoferrin, it says the structural genes. It has Z, Y, and A, the three genes we've already discussed, that is beta galactosidase, permease, and transacetylase. And talking about the promoter, it is the binding site for RNA polymerase to initiate transcription of structural genes. So, your structural genes, that is Z, Y, and A, they will be transcribed when RNA polymerase is going to read them, and the entry of the RNA polymerase is through binding to the promoter site. Now, the operator region, this is a regulatory region near the structural gene that is downstream to the promoter. It controls access of RNA polymerase to the structural genes. Okay, how is it controlling? Because this is operator region is the region where your repressor protein is going to bind. So, your RNA polymerase is under strict regulation. Now, there's another regulatory gene, I. This encodes the lac repressor protein. So, your lac I gene, it is going to produce lac repressor protein, and this is a constitutively expressed gene. That is, it is expressed all the time. It produces a repressor that binds to the operator, inhibiting transcription. Okay, so this is that negative regulator of RNA polymerase. Here in this diagram, you can see here, I've made this. Okay. Now, the regulation mechanism in absence of lactose. So, these are the various conditions that I've already discussed. Let's read them. So, in the absence of lactose, what is happening? The lac repressor binds to the operator, preventing RNA polymerase from transcribing the structural genes. Okay, because why? Allolactose will not be present. Okay, in the absence of lactose. So, your allolactose is absent. And what was the function of allolactose? I told you that when lactose is present, small amount of lactose is able to enter the cell, change into allolactose. This allolactose is going to bind to the repressor protein at the operator region, and it is going to bring a transformational change in the repressor protein, as a result of which the repressor protein will be pulled off or pulled away from the operator region. So this was happening in the presence of lactose. But since this is the scenario in which the lactose is absent, what is going to happen? Repression occurs when lactose is not available for metabolism. And this repression is removed by allolactose, like I just explained, in the presence of lactose. So in the presence of lactose, now this is the scenario. Lactose acts as an inducer. Okay, lactose binds to lac repressor, causing a conformational change that makes it unable to bind to the operator. Now, RNA polymerase can now transcribe the structural genes, leading to the synthesis of enzymes for lactose metabolism. Now, positive regulation. So, this just the removal of the repressor protein from the operator region is not enough. You also need what? You need positive regulation as well. So you also need the push. Okay. So these two conditions, both of them should be satisfied. Okay. Now cyclic AMP is a signaling molecule. Cyclic AMP binds to catabolite activator protein, that is the CAP protein. Now CAP, cyclic AMP complex, binds to a site near the lac promoter. That site is known as CAP region. Okay. It enhances the affinity of RNA polymerase for the promoter, increasing the transcription. Now, talking about the significance, lactoferrin allows efficient use of resources by only producing enzymes needed when lactose is present, demonstrates the concept of gene regulation responding to environmental conditions. So, this is how your E. coli is able to respond to environmental conditions. That is, in different scenarios. That is, first scenario can be your glucose present, lactose absent. Second scenario, glucose absent, lactose present. And third, both glucose and lactose present. So depending upon the environmental conditions, it is going to switch on or off its lactoferrin. Okay. Now the last topic. Explain regulation of gene expression in eukaryotes. Now, regulation of gene expression in eukaryotes, it is brought about by different mechanisms. On each of these mechanisms headings, they themselves are like an entire chapter in themselves. So, I'll briefly explain you all of these. Transcriptional control. So, promoters and enhancers, we've just seen the promoters and enhancers. Here in this case, it was your enhancer was your cap region and the promoter was where the RNA polymerase was binding. So DNA sequences regulating RNA polymerase binding. They are your promoters and enhancers and transcription factors. Proteins modulating transcription by binding to DNA. So the various transcription factors. Then histone modification. These are chemical changes to histones affecting chromatin structure. So that is transcriptional control, then followed by epigenetic modifications. Now, what is epigenetics? Okay, epigenetics literally means like from outside or above. Okay, so these are like reversible hereditary chemical modifications in DNA or chromatin. Okay, so your epigenetics, these are reversible hereditary chemical modifications in DNA or chromatin which do not change the nucleotide sequence. Okay, now this is important. So these are like from the outside. Okay, for example, if you see a caterpillar then changing into pupa and then it changes into a butterfly. So the underlying genetic composition of all the three is same, okay, but the morphology is entirely different, that of a caterpillar and the butterfly. But but because of the epigenetical modification, what is happening? Your caterpillar is entirely different from the butterfly, although the genes are remaining same. So this is something that is happening from the outside or above, okay? So these are reversible. They are not like your mutations, which are irreversible. These are reversible hereditary chemical modification, and they do not just take place in the DNA, but also to the histones. Okay, therefore it says chromatin. Okay, because your histones are present in the chromatin, which do not change the nucleotide sequence. So that is your genetics. Now these epigenetic modifications, they can be of two types: either DNA methylation or demethylation, that is addition of methyl groups to the DNA, or they can be histone acetylation or deacetylation. Now what is the effect of DNA methylation? That is when you're methylating a DNA, what happens is it leads to gene silencing. Okay, so your DNA methylation, it leads to gene silencing. And your histone acetylation, what is acetyl? Acetyl is having negative charge and your histones are positive charge. So your DNA is a negatively charged molecule and they are wrapped around histone molecules which are positively charged. So the negative and the positive charge, it is able to condense the DNA. Now what happens when histones, they are bound with acetyl, so the positive charge of the histone is decreasing, as a result of which the DNA becomes less condensed or it becomes euchromatic. So histone acetylation is going to increase what? It increases euchromatin. Euchromatin is that form of DNA which is less condensed. And I just explained you the mechanism that why it becomes less condensed, because histones are positively charged and acetyls are negatively charged. So when acetyls, they bind to histone, the positive charge on the histone is reduced. So therefore, the DNA becomes less condensed. So your histone acetylation is basically increasing the euchromatin. And because of less condensation or because of increased euchromatin, your promoters get open or exposed. So basically, in short, if you try to remember, your histone acetylation, it increases or it activates genes. So it leads to gene activation. So that is the effect of DNA methylation and histone acetylation. So histone acetylation, it is addition or removal of acetyl groups to the histones. Now RNA processing, it is brought about by alternative splicing in RNA editing. Now what is alternate splicing? So you know what is splicing? Removal of introns and splicing or joining together of the exons. Now for example, this is exon 1, 2, 3 and 4. Okay, the introns have to be removed. These are introns in between. Okay, so now one way is that all of the exons, they are spliced together. That is 1, 2, 3 and 4. This is one way. The other way can be exon 1, exon 2 and exon 4. They are spliced together. A third way can be exon 1, exon 3 and exon 4, they are spliced together. So you see, from the same mRNA, you're going to produce different proteins. So this is alternative splicing, okay? Div Produces different mRNA isoforms by selectively including, excluding exons. So they are going to select which exons are supposed to be spliced together. So out of the four exons in the first, all the four exons are spliced together. In the second, you are splicing 1, 2, and 4 exons. In the third case, you are splicing 1, 3, and 4 exons. Okay, so this is what is known as alternative splicing. Now, what do you mean by RNA editing? This can be understood with the example of protein ApoB48 and ApoB100. Okay, so ApoB48, this is produced in the small intestine, and ApoB100, it is produced in the liver. So what is happening in the intestine because of spontaneous changes? Your cytosine is changing into uracil. Okay. So cytosine is undergoing spontaneous deamination and it changes into uracil. As a result of which a translational stop codon is introduced. And your translation is arrested. So only 48% of the mRNA can be translated. Okay. So therefore, in the small intestine, ApoB48 is being formed. This is known as RNA editing. And whereas in the liver, the entire mRNA is going to be translated, 100% of it, and therefore the protein produced is ApoB100. So that was about RNA editing. Now, post-transcriptional regulation, it is brought through miRNAs and siRNAs, which is involved in gene knocking and gene silencing. Then RNA stability influencing mRNA half-life through regulatory elements. Then talking about transitional control. So initiation factors, these are proteins regulating the start of translation. And RNA binding proteins, these interact with mRNA to influence translation efficiency. So either you can control the translation. Then post-translation modification, that is through phosphorylation or dephosphorylation, covalent modifications to protein affecting stability, activity, or cellular localization. Then cell signaling pathways, cell surface receptors, and intracellular signaling cascades. That is one way through which you can control the expression. Then hormonal regulation being the last, that hormone receptor interactions and endocrine system. So through hormones binding to receptors, you can also influence gene expression. So that was regulation of gene expression in eukaryotes through various mechanisms. So the important ones I've discussed. So with this, we come to the end of today's lecture. That was regulation of gene expression. So if you have any queries, you can ask. Thank you for watching. See you in the next class.